Welcome to Planetterrian Daily, episode 136, for July 30th, 2026. Here's your science and health briefing. Nanoplastics reach the neonatal mouse brain and form distinct spatial patterns visible in 3D. Researchers used three-dimensional imaging to map the distribution of nanoplastics smaller than 1 micrometer inside the brains of neonatal mice. The particles enter through environmental exposure routes including food, water, and air before accumulating in specific brain regions. The work shows that determining exact locations within complex organs has been technically difficult until now. The method reveals how degradation products from everyday plastics interact with developing neural tissue. Future studies will track longer-term clearance or retention of these particles. The three-dimensional mapping of nanoplastics in neonatal mouse brains shows particles accumulate in localized patterns rather than spreading uniformly. This spatial information helps explain why developing neural tissue may be particularly sensitive to environmental plastic fragments. The imaging approach can now be applied to track particle clearance or persistence over longer periods. It also provides a baseline for testing whether specific particle sizes or surface properties alter distribution. Now shifting from environmental particles reaching the brain, we move to how genes themselves are buffered against disruption inside developing cells. The study focuses on TCOF1, a regulator of RM synthesis whose reduced dosage causes Treacher-Collins syndrome. Transitional cranial neural crest cells show extreme sensitivity to TCOF1 levels during mesenchymal specification, while RNA polymerase I itself does not trigger the same response. CNCC-specific enhancers stabilize expression when promoter regulating factors fluctuate, converting variable outputs into threshold-protected ones. Experiments pairing promoters with enhancers across multiple housekeeping genes confirm the buffering principle operates broadly. The mechanism explains why certain broadly expressed genes produce tissue-specific developmental disorders. While enhancers protect gene dosage, another layer of control operates at the tissue level through physical cell junctions. Many tissues rely on continuous stem cell division to replace cells. Yet unchecked proliferation disrupts homeostasis and can promote tumors. The research identifies cell junction proteins as regulators that restrain stem cell activity under normal conditions. Loss of this control allows excessive division that alters tissue architecture. The mechanism operates across multiple animal tissues. Future work will test whether restoring junction function can reestablish balance in disease models. Most people picture stem cells as constantly ready to divide and repair tissue whenever needed. In reality, many tissues keep these cells under tight local control so they do not over-proliferate and disrupt normal structure. Right now, proteins at cell junctions are monitoring stem cell activity in your skin, gut lining, and other renewing tissues, releasing the brake only when replacement cells are actually required. One memorable detail is that losing this junction-based restraint can shift the balance toward uncontrolled division within days in experimental models. The next time you notice a cut healing without excess growth, that outcome reflects these local checks doing their job. Researchers are now testing whether strengthening junction signaling can restore balance in conditions where stem cell activity has escaped regulation. Watch for studies that measure junction protein levels in human tissue samples from aging or chronic injury settings. From cellular restraints inside tissues, we turn to a materials-level discovery that also imposes order through mechanical forces. Researchers at the Max Planck Institute for the Structure and Dynamics of Matter and the University of Oxford applied strain to piezomagnetic and piezomagnetic materials. The stress generated electrical polarization or magnetization and simultaneously produced left or right-handed chirality on demand. The effect opens a route to control chiral electronic properties through mechanical input. The work appears in Nature. Follow-up experiments will test whether the induced chirality persists after strain is released. Shifting from physical materials to biological materials in the ocean, hydrothermal flows create their own ordered communities. The German research vessel Meteor located a previously unknown hydrothermal system near the Greek island of Milos at 100 to 250 meters depth. Different intensities of fluid flow produced distinct microbial communities and controlled mineral formation on the ocean floor. The findings demonstrate how hydrothermal activity structures both biology and geology at intermediate depths. Samples were collected during Expedition M192 in 2023. Further mapping will determine how widespread such systems are in similar settings, from seafloor systems to terrestrial ones. We look at how death itself structures ecosystems. An international team led by the University of Granada argues that processes following animal death have been understudied in ecology. Carrion affects nutrient cycling, scavenger communities, pathogen transmission, and potentially early human behavior around carcasses. The perspective draws on evidence from multiple continents and time periods. It proposes carrion as a unifying factor across ecology, epidemiology, and anthropology. Expanded field studies will quantify these links in contemporary landscapes, from ecological processes to historical human management of animals under environmental constraints. University of Michigan researchers analyzed isotopes in sheep molar enamel from a medieval town site in arid Kazakhstan. The isotopic signatures indicate herders adjusted grazing and water sources seasonally to cope with limited rainfall. The data show fine-tuned strategies rather than uniform practices across the region. The approach links animal management directly to environmental constraints of the period. Comparable studies at other Central Asian sites are planned. New findings on particle distribution, gene buffering, and tissue regulation add concrete mechanisms to ongoing questions in environmental health and cellular maintenance. Keep an eye on expanded mapping of shallow hydrothermal systems and their microbial signatures in the coming months. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcast or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. Quick tip from the network. Try environmental intelligence next. The Environment and Climate Policy Brief for Canada. Every NARA Network show is free at naranetwork.com. And on the website, every episode's visuals live in our free image gallery at naranetwork.com/gallery. Royalty free under Creative Commons. Download with attribution. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.